Hello everyone, this is Latha Akula. In this video we are going to study about the remaining part in the Meiosis-1. In the last video we have completed the Prophase 1. In that we have studied the 5 stages. So in that 5 stages what happened? Between the homologous chromosomes, the exchange of genetic material occurs, that is crossing over completed. And that is nothing but recombination of the genetic material takes place by means of an enzyme recombinase. At the same time, in the Prophase 1, at the end of the Prophase 1, in the diakinesis, the nuclear membrane around the chromosomes also disintegrates. Now, the cell enters into the Metaphase 1. So, in the Metaphase 1, what will happen? The chromosomes move to the cytoplasm freely because there is no boundary for the chromosomes because the nuclear membrane completely disappeared, degenerated. Then the chromosomes they freely moves in the cytoplasm, and these chromosomes they align themselves in the equatorial plate. So this is the homologous chromosomes pair. This is one homologous chromosomes pair. These chromosomes they align themselves in the equatorial plate. Here this is one equatorial plate and this is another equatorial plate, and these homologous chromosomes arranged in two rows. They were arranged in two rows and form double metaphasic plate. In the metaphase 1, what will happen? These homologous chromosomes align in two rows and forms the double metaphasic plate. And now after completing this metaphase 1, the cell enters into Anaphase 1. Here what will happen? The chromosomes which have been attached to the spindle fibers that was pulling the chromosomes to the opposite ends. The homologous chromosomes pair was separated here. So what is happening? The spindle fibers, the length of the spindle fibers that will be short ends, and the chromosomes, this is a homologous pair, right? So in that pair, one chromosome moved to one end, another chromosome moved to the opposite end. Like that, these homologous chromosomes, they were separated and moves to the opposite ends. Next, they starts moving towards the poles, and these homologous chromosomes pair only one chromosome was reaching the opposite pole and another chromosome was moving to the another pole. That means what is happening here? The number of chromosomes becomes half. Actually, we have started initially the meiosis started with 4 chromosomes, but now what is happening? This is one cell and this is another cell, right? Here only 2 chromosomes was moving to the one end and 2 chromosomes moving to the another end. That means the number of chromosomes were reduced. The reduction in number of chromosomes started in the Anaphase 1. Next, the cell enters into the telophase 1. So what will happen here? In the telophase 1, the complete disintegration of the spindle fibers they disappears, and the nucleolus and nuclear membrane that starts reappearing. That means the nuclear membrane is formed around the separated chromosomes, and at the same time nucleolus also reappears here. And then these chromosomes around these cells, the cytoplasm also formed, and that means the cytokinesis also occurs. After cytokinesis, what we will study? Here the 2 daughter cells were formed. From the meiosis 1, how many daughter cells were formed? 2 daughter cells were formed, and which have the half the number of chromosomes. And after cytokinesis, what the cell will do? The cell will enter into the interkinesis. But here, what will happen is the chromosomes within the interkinase, when the cell enters into the interphase, the chromosomes that starts decondensation. That means the chromosomes will be released and the loose thread-like structure will reappear. That means chromatin is again formed. But here what will happen? The chromosomes they partially uncoil, but they remain straight like the chromosomes only. That means here what will happen? The chromosomes will not enter into the extremely extended interphase. And these 2 daughter cells, they have a single nucleus. This is having one nucleus and this is having another nucleus. And with one chromosome from the homologous pair. This is homologous pair, right? We have studied. So in the homologous pair, one chromosome enters into one cell and another chromosome enters into the another cell, and this is another homologous pair. Here also the homologous pair have been separated. The separated chromosomes reaches to the two daughter cells. Here the number of chromosomes reduced to half, but they have the double amount of DNA is present. Both the chromatids were moved to the opposite end. Here the separation of these chromatids takes place in the next coming meiosis-2. So after completion of the meiosis-1, when the cell entering into the meiosis-2, there is a gap is present which is called as interkinesis. So after completion of the telophase 1, the cell enters into the cytokinesis, where the cytoplasm was separated between there is a constriction is formed and the cytoplasm was separated, and then two daughter cells were separated, and each daughter cell has its chromosomes, half the number of the chromosomes. That means that each daughter cell was in the haploid condition. So from the meiosis-1, how many daughter cells were created? Only 2. And what is the condition of these cells? In the haploid condition. And what we have initially started for the meiosis? We have started with diploid cell and we are getting the haploid daughter cells. And after completion of this, the cell it has to enter into the meiosis-2. But between the meiosis-1 and meiosis-2, there is a short gap is present. That gap is called as interkinesis or intrameiotic interface. Interkinesis or intrameiotic interface. Generally, in the interface, what will happen? The chromosomes, the structure of the chromosomes will be uncoiled, and then the chromosomes will form the chromatin. Right? Generally, that will be happen. But here, the chromosomes elongate, but there is no chromatin formation. The chromosomes will be in the they were just straight only. There is no chromatin formation here. And at the same time, in the interface, what will happen? DNA replication also occurs. But initially, before meiosis-1, in the interface, only the DNA replication occurs. But here in this interface, there is no DNA replication occurs. And if the cell is in animal, if it is animal cell, the centrioles are required, right? The duplication of the centrioles also occurs in the interkinesis. And not only that, RNA and proteins which are required for the next coming meiosis-2 were also prepared in the interkinesis. This is all about the meiosis-1. So how many daughter cells were formed here? Only 2 daughter cells with half the number of the chromosomes were present. Now in the next video we are going to study about meiosis-2.